Okay, welcome everyone to the second presentation over the last couple weeks devoted to the Lehmann family. I appreciate those who are joining live tonight, and I will be posting this to YouTube. But last week in the first presentation we talked about the documentary evidence for the Lehmann family that exists from the town of Weiderstadt in Hesse, Germany, and I think it was a really good discussion. And we saw records going back to as early as 1780 for our Lehmann line. Today we're going to view things from a different lens, from Y-DNA. And we're going to go back thousands of years looking at the Lehmann paternal line and learning more about that history. We're going to touch on some other aspects of human history as well in this presentation that are pretty interesting. So we're first going to start with some terminology, and then we will get into the testing and the results. All right, so why DNA testing? This slide provides an overview. What is a Y-DNA test? So a Y-DNA test is a DNA test that can only be taken by males. And why is that? It's because it looks at a male's Y chromosome. Only men have a Y chromosome, women have two X's, men have an X and a Y. And that Y chromosome that males have are inherited from their fathers. And so a Y-DNA is looking strictly at this direct paternal line from father to son, father to son, father to son through the generations. Now, because Y-DNA mutates very slowly, we can trace migration patterns and learn about human history over long periods of time by studying Y-DNA. I have a slide here that discusses Y-DNA versus autosomal DNA. So, what we're going to be discussing tonight is predominantly Y-DNA, and it is very different from autosomal DNA. So what, again, is Y-DNA? We've talked about it. It is looking at the paternal line of males that they inherit through their Y chromosome, and it's passed down from father to son. Now, this Y chromosome is passed down almost entirely intact from father to son, and it allows us to look back thousands of years when looking at a Y-DNA test. When we talk about Y-DNA, we say that it's a narrow but very deep test. It only looks at a very small sliver of one's ancestry, that direct paternal line, but because it gets preserved, the Y-DNA gets preserved over time, we can look back thousands of years. Now let's compare that, and that's on the left-hand side of the page here is that Y-DNA graphic. Now if we look to the right-hand side, we have autosomal DNA. And autosomal DNA is what most people think of when they think about DNA tests. An autosomal DNA test looks at the 22 pairs of autosomes. You have 23 chromosomes. 22 of them are autosomes that are inherited 50% from each parent. And so you are able to see all lines of your ancestry with an autosomal DNA test. You can see here with an autosomal DNA test, you will get 50% from each parent, 25% from each grandparent, 12.5% from each great-grandparent, and 6.25% from every second great-grandparent. So you can look at all lines of your ancestry, but if you think about those percentages, the deeper ancestry gets diluted over time. You are only going to inherit a small sliver from these earlier ancestors. And so even though you can look across all lines of your ancestry, because it gets diluted, autosomal DNA can only really provide insights within the past 200 to 400 years. You can't really go further back in time than that because of that dilution. Your genetic ancestry is going to be much smaller from these earlier ancestors, or you might not actually inherit your autosomal DNA from some of these ancestors as you go further back in time. So the way to think of autosomal DNA, that has a broad but a relatively shallow reach in comparison to the Y-DNA, which is narrow and deep. And autosomal DNA Those are the big providers of DNA testing that you think about today, whether that is Ancestry or MyHeritage or 23andMe. They are the main providers of autosomal DNA tests. For Y-DNA testing, there's really only one main commercial company that does this extensive DNA testing for Y-DNA, and that is Family Tree DNA, and they are based in Houston. Okay, so we're going to go into a little bit more about Y-DNA terminology. This is the most complicated slide from a terminology standpoint, but if you are able to understand the concepts here and this little illustrative graphic, it will help a lot. It'll go a long way to understanding Y-DNA and how it works. So we're going to start off by talking about haplogroups. So a haplogroup is a way to describe the deep ancestral history of a paternal line, and it's defined by mutations in the Y-DNA. So what is a haplogroup? It's a genetic population group of people who share a common paternal ancestor because they all share the same set of mutations on the Y chromosome. So mutations are also called sometimes SNPs, single nucleotide polymorphisms. So oftentimes in the literature you'll hear SNPs. But in this presentation, we're going to talk about mutations. Now, what are mutations? It's really, there are point mutations in the Y chromosome where a letter gets changed. Instead of having a C, you have a G. Instead of an A, you have a T in that one point in the Y chromosome. And they don't happen very often. Usually when the Y chromosome gets passed from father to son, it passes down almost entirely intact. So everything is exactly replicated from father to son. But every 50 to 150 years, so every few generations, there is one small change that happens when it gets replicated from father to son, and a new mutation forms. These mutations, I want to be clear, they are not negative. There's no negative connotation with them. It's just a neutral thing that happens where in the replicating of the Y chromosome that gets passed down, it just so happens that a letter was copied incorrectly and it got changed. It doesn't have any negative impacts in terms of, physiologically or health-wise. There are some mutations that can occur on the Y chromosome that can affect spermagenesis. But when we're talking about these mutations here in the Y chromosome, they're just neutral. They're just literally changes that occurred every 50 to 150 years, and they're markers of time. And so, haplogroups, as I just mentioned, they really mark periods of time when your group branched off by having a new mutation form. So the major haplogroups, so those are the ones that formed really early on. They are given letters of the alphabet between A and T. And then the subgroups, which there are many different subbranches within all these letters between A and T, they are called subclads. So we're going to talk about subclads. And in essence, the haplogroups are these giant Russian nesting dolls in which they're increasingly narrow and smaller Russian nesting dolls. They're smaller subclads that are found within these haplogroups that mark different branches of that line. So maybe it helps to give an example. So this one on the right-hand side is an illustrative example of haplogroups. So at the top here, you have the earliest ancestor who doesn't have any mutations. If you go back early in time, there's a concept of the Y chromosomal atom, of the first male who all humankind today descend from. That man lived about 250,000 years ago in Africa. So we can think of, at the very beginning, there was this Y chromosomal atom who didn't have any mutations. And then this person had two sons. The first son was his DNA, Y DNA, was copied exactly the same. There's no mutations that formed. But the second son had the red dot, and that's signifying a mutation. And so at this point in this new generation, we now have two haplogroups. This first one at the top, which is intact, that is what we call A. And this red dot one is haplogroup B. It branched off into two different haplogroups. And haplogroup B will always have this red dot in their Y chromosome, and it'll get passed down for thousands of years. Everyone will have this red dot, which signifies a mutation in the Y DNA. And then we can take it one more generation, and we're looking here at the next generation, and now we see another set of mutations that form. So the person who didn't have any mutations, he has one son who still has no mutations, but he has another son who has this green dot. And this green dot forms a new subclad, a new haplogroup in this case, a new major haplogroup, and it's haplogroup C. And likewise, the other son with haplogroup B, he has two sons. One son is entirely intact and just has the red dot, and he has a second son who has the red dot and the blue dot, which forms a new haplogroup D. So when we're looking today in the modern day population, all we can see are the people who are closest to the present day, these people at the very bottom. And we can look at their mutations that they have, and we can see who is more closely related to each other. So looking at this bottom row, we can see haplogroup D is closer related to haplogroup B than it is to C or A. Why is that? Because both D and B, they both have that red mutation. So they must have a common ancestor that's before the A and C. And likewise, because C doesn't have the red dot, it is more closely related to A. And so we try and make these inferences based off of the modern day population. But we're also going to talk about this new field that's taken place over the past decade, which is finding DNA in ancient people. And Y DNA has also been a big factor in that. And we can actually look at ancient Y DNA to get insights about what people were like back in ancient history. But most of the Y DNA research does look at the modern day population, and it looks at what mutations people have today to infer things about the past. Now, I talk about this Russian nesting doll, and sometimes it can get confusing for people because we're going to talk about a lot of different subclads today. And, when people are looking at Y DNA, oftentimes they're thinking, oh, I'm just this, versus having a more inclusive view. So a way that I like to describe this now is a nice analogy is to talk about the question, where do you live? If you were to answer, where do you live? There are a lot of different answers you could give. So if you wanted to give a very broad answer, you could say, well, I live on planet Earth. And for the vast majority of people today who aren't, for the few people who aren't at the International Space Station, that would, consist of almost the entire population. So when you say, oh, I live on planet Earth, that's pretty similar to talking about the Y chromosomal atom, where everyone descends from the same person. But then you can get a little bit more specific, right? You can say, well, I live in the Northern Hemisphere. And well, that's a little bit more specific, but the majority of the world's population lives in the Northern Hemisphere. You're still not getting all that degree specific. But then, you could say, well, I live in North America. Okay, well, that gets a little bit more specific. You could then get even closer and say, well, I live in the United States. And you could take it further and say, well, I live in the state of New York. And you see, as we go through each of these steps, you're getting narrower and narrower. You're going to share that location with fewer and fewer people. And it is analogous to getting closer and closer in time to the present day in Y DNA language, right? So we can take it further from the state to the county level, to the city, to the neighborhood, to the street, and then maybe even to the same house. So we're going to try, and you can, say that I live in this neighborhood, but that also means you live in this town. It also means that you live in that county and in that state. It's inclusive of all of these. So when we talk about Y DNA tonight, we're going to start at a very broad level, talking about the haplogroups, and we're going to get, and that's very early, very ancient, going back thousands and thousands of years. And we're going to slowly progress closer to the present day. But we are members, of all of these groups. It's just they're markers of different periods of time. We're going to start back, at the ancient past, and we're going to get closer to the present day as we proceed. So I'm going to stop here, and if anyone has any questions, I'm happy to take that before we continue on, because I know this is a lot of terminology. All right, I think we can continue on then. All right, so now let's get into the y-DNA testing. Okay, so as you probably already know, I've always wanted to learn about my ancestry, and I started my research with the Lehmann paternal line. And you can see here on the left-hand side, this is my paternal line, starting from me and then going to my father and grandfather, all the way back to my fifth great-grandfather, who we talked about last week, Abraham Isaac Lehmann, who was born in the mid-18th century. 1741 might be a little early, but certainly by 1750, 1755, he was born. And so, for me, I wanted to explore the research from a y-DNA angle. And, fortunately for me, this is not usually the case when I study other surnames, right? But because I am a male and I have the Lehmann surname, I could do y-DNA testing myself to learn about the Lehmann line. And that's what I decided to do. So what can y-DNA testing help answer? Well, some questions that it can help answer are: where did the Lehmann paternal line originate thousands of years ago? Which other y-DNA testers are connected to the Lehmann paternal line, and where is their ancestry from? So it's looking at other modern day testers and seeing how closely connected they are to the Lehmann paternal line. And then the last one, and we're going to touch on this on the second half of the presentation, is, I have found documentary evidence of the Lehmann line going back to this town called Weiderstadt. But can I find y-DNA evidence of this as well? Is y-DNA supportive of the documentation that our Lehmann line is biologically from the Lehmann family in Weiderstadt? So back in 2020, so this is almost 5 years ago, I took a y-DNA test, and then I upgraded it to the extensive big Y test later in 2020. So this was a few years ago when I initially took this y-DNA test. All right, so this is sort of the headline slide talking about the different haplogroups and subclads that we're a part of. And again, with each one, we're going to get closer to the present day. This is just an overview, and we're going to go through on each slide, going through each of these pieces. So initially, we are part of major haplogroup G, and that haplogroup formed a long time ago, about 46,500 BCE. So almost 50,000 years ago is when haplogroup G formed. Within haplogroup G, so now we're getting a little bit closer to the present day, we descend from this subgroup, a subclad called GM377, and that formed about 10,000 BCE. And then within that, we're going to talk about GBY794. That formed in 2700 BCE, so we're getting closer again to the present day. Within that, we are part of GBY764. We're going to talk a lot about that because this line formed about 750 CE, this subclad, and it's entirely Ashkenazi Jews. So everyone who's tested within that is Ashkenazi Jewish along their paternal line, and so we'll talk more about that. And then within that, we're in a subclad of the Ashkenazi Jewish group called GFT7446 that formed in 800 CE. And then finally, the closest that we could get to the present day with my testing was the last haplogroup, which is GFTA54806. So we're going to go through, and that formed around 1200 CE. So we're going to go through each of these different things, and we're going to learn a little bit about our line and the ancient history associated with it. Okay, so I always include this little timeline of human history and then sort of plug in the different the haplogroup and the major subclads that we descend from, just in the context of human history. So G formed very early on. Again, this is like almost 50,000 years ago. It's after the first abstract art was created, but it's older than the oldest figurative art. We then get to GM 377. This is at the beginning of the Neolithic era, so it's coming out of the Ice Age. And then finally, and then next we're going to do BY 794. This is in a time where we do have, writing in human history in certain parts of the world. This is 2700 BCE. But it is before the formation of Judaism. And then we fast forward to BY 764, the Ashkenazi Jewish clad, and this is really the beginning of the Middle Ages, where the early Middle Ages is when that formed. And then finally, in the middle of the Middle Ages, in 1200, we have GFTA 54806. So again, we're going to go into all of this, but I like this chart that shows the context of human history within all of these groups that we're a part of. All right, so we're going to start from the beginning here with major haplogroup G. So that's the major haplogroup that we descend from. As I mentioned, it formed around 46,500 BCE, and most likely the early ancestor was living somewhere in the Near East. So haplogroup G, before haplogroup G, it descends from macro haplogroup F, and F was that earlier ancestor to G, where it represented the second major migration of Homo sapiens out of Africa at least 60,000 years ago. So haplogroup G formed after the earlier ancestor had left Africa and had come to the Near East, and that's where haplogroup G formed. Haplogroup F, we know, came to the Arabian Peninsula and settled in the Middle East. So if we wanted to take this further, we also descend from haplogroup F, but we broke off of that and formed haplogroup G. So the main branch that descends from macro haplogroup F is called IJK. And 80% of modern Eurasian people descend from IJK. So we descend from the other part, the minority of people who lived in Eurasia, who broke off from it. So haplogroup G is a side lineage. It evolved in isolation from other people for tens of thousands of years, and it was cut off from the waves of colonization of Eurasia. So it basically stayed in the Near East and was isolated and was not involved with settling other regions of the world, like some of these other ones. Like IJK, they end up settling in different parts of the world, whether it's in East Asia or whether it's in Europe. G sort of stays in the Near East. So where do we see people from haplogroup G today? It is still, a good amount of people in the world. You can find them from Western Europe and Northwest Africa to Central Asia, India, and East Africa. But they're generally at pretty low frequencies. So for most places in the world, only between maybe 1 to 10% of the population might be in haplogroup G. And again, haplogroup G is huge, right? We're going to talk about lots of different subgroups within haplogroup G. Haplogroup G is a major haplogroup, but it's still in very small percentages from people in these places of the world. The only exceptions for haplogroup G are in the Caucasus, southern Italy, and Sardinia, where between 15 to 30% of male lineages are in haplogroup G. So again, the Caucasus here and in some parts of southern Europe, Mediterranean Europe, that's where we see the highest concentrations, but it's still between 15 and 30% of men in just those places. So we're going to talk about this, but haplogroup G has two main branches, especially G2, which is the main branch of G. There were two main sub-branches. The first one is GP15, which is also known as G2A, and the second one is GM377. We're going to actually talk about both today. We are descended from the second one. The Lehmann line is descended from GM377. But I do want to talk about G2A and GP15 because it's an important part of human history. So we're going to start with actually G2A. So that GP15 that I mentioned, we do not descend from this branch. In fact, our branch branched off from the G2A. We don't share a common ancestor until 20,000 years ago. Or actually, further than that, 20,000 BCE, 22,000 years ago, okay? So we don't share a common paternal line ancestor with this group, G2A, until you go back 22,000 years ago. So why am I going to talk about G2A? Well, first of all, it's really interesting. It plays a really important role in European history. So that's number one. And I want to discuss some of this in this presentation because it's a good time to talk about what we know about G2A. The second is it's a good contrast to contrast what we know about G2A and what we know about R line, which is part of G2B. Because there is a big disparity in our knowledge between these two different branches. And then the last one is, guess what? We also descend from G2A and other branches. So this is part of our ancestry that is worth talking about, but just not through the Lehmann line. Why do I say that? Well, for those people on the call, anyone who descends from Grandma Liz's mother's family, the Stein family, they are G2A males, okay? So our Stein family, this is their history. This is their paternal line history. They branched off of the Lehmann line about 20,000 BCE, but this is actually their history of where they descend from, and this is their history. So I think it's worthwhile to talk about from that angle and to talk a little bit more about what we know about G2A. So it's not talking about the Lehmann line because this line branched off 20,000 BCE, but it's interesting, and again, we do have branches of our family that are known to be part of G2A. Okay, so let's talk about G2A. Again, we share that common ancestor from 20,000 BCE. G2A formed about 18,000 years ago during the last glacial maximum. So what do we know about G2A? Well, what's very interesting is G2A is closely linked to the development of early agriculture. So agriculture, as we know it, began, about 11,500 years ago in the Fertile Crescent. That we know that it formed there, and there were a few haplogroups that were associated with that. If you want to check out the Rubin Y-DNA presentation, we've talked about the Natufians from haplogroup E. They were among the earliest to do early agriculture. But G2A is a haplogroup that is associated with agriculture's expansion into Anatolia, the Caucasus, and then into Europe. So when we look at ancient DNA today and trying to see which men in ancient cultures were part of G2A, we don't really see so much of that in the Levant. But we do see a lot of it in Anatolia and then spreading out into Europe. And in fact, G2A was the dominant lineage of Neolithic farmers and herders who migrated from Anatolia to Europe between 9,000 to 6,000 years ago. So you can see this migration going from the dark red all the way into the yellow and green. This is the spread of agriculture into Europe. And what we know is that these early European farmers, they came originally from the Fertile Crescent and then came to Anatolia, and then they spread westward, bringing farming and agriculture to Europe. Before then, everyone was just hunters and gatherers that lived there. And in these cultures, we see the innovation of agriculture being brought westward into Europe over a period of 3,000 years. G2A is the dominant lineage in these early Neolithic farmers. So it's the main line that we see in paternal lines, spreading from northwestern Anatolia, starting in 6,500 to 6,200 BCE in Greece, and then into the Balkans, and it spreads westward into Europe. And most Neolithic cultures in Europe that have done Y-DNA analysis, the majority of men are these G2A men who originally came from Anatolia. Examples of it here, I have some links here to some of these early cultures, ancient cultures, in Hungary and Croatia and in Germany and in southern France and Iberia. All of these places have G2A men who originally came from further east. It's interesting that G2A remained the principal paternal lineage even when farmers intermingled with the indigenous populations. The G2A men retained they retained the majority of the male population in these cultures. All right, so I like to include this map here. So again, in the last 10 years, there's been all of this research into ancient DNA and being able to take samples from bones, especially like the inner ear of skeletal remains, and being able to extract DNA. And they've been able to do it not just for Y-DNA, but also autosomal DNA. But, from here I want to just talk about the Y-DNA. So this is like a map of all the different ancient DNA sites where there are G2A men, and they are dated. So some of the earliest ones are here in Anatolia, and then you can see them spreading into Europe and spreading into Iberia. And in all of these places are these G2A men. So I think this is a nice map, and this tree will get updated to this website as more ancient DNA are found, but it was found throughout Europe. All right. So I want to spend some time talking about what happened to these early European farmers that came from Anatolia and G2A, because unfortunately it didn't have a great ending for these populations. So although G2A was an important paternal lineage during the Neolithic era, it's far less prominent today in Europe. So if you look at the modern day population as opposed to the ancient population, you're going to find G2A very sparsely. There are not that many men that have G2A today, and they're mostly found in mountainous regions of Europe. And this is actually linked to a very, very important finding. There was a landmark paper. I mean, it's such an important paper that came out in 2015 that was talking about a massive migration that took place into Europe that was discovered and supported through DNA analyses. Okay, so what happened? Well, there was an ancestral population which have been termed the Proto-Indo-Europeans, and they originated in the Pontic steppes of Europe. So this is in places north of the Black Sea, in modern-day Ukraine today, in eastern Ukraine, spreading out into parts of southern Russia. There was a population that came out of there and who invaded Europe. And we know that over the next few hundred years, so they left Europe, they left the Pontic steppe around 3300 BCE, and this culture was called the Yamnaya. Why are they called Yamnaya? Well, they were named by a researcher. They're marked by burying their deads in these kurgans, which is a burial mound, and inside there would be these pit chambers where they would bury their population, probably their leaders. And Yama means pits in, I think, Russian. So the name of this culture was given Yamnaya, and they came westward into Europe between 3300 BCE, and over the next few hundred years, they basically destroyed much of the existing population that existed in Western Europe. Now, there are some questions of exactly how this happened, because there definitely were invasions of some sort. They were a militaristic population. But there have been also some other supporting factors that may have led the Yamnaya to be so successful. So there was a recent paper that came out, I think earlier this year, that looked in places in southern Scandinavia, and before the Yamnaya came, they found burial grounds of people who were infected with the plague. This was an earlier form of the plague before it became as bad as it was in the Middle Ages, but it wasn't as bad, and it couldn't be infected from fleas. But there was an earlier version of the plague, and there was huge percentage of people buried in these, and this has preceded the Yamnaya coming, who had this plague and probably died of it. So there is a thought that there was a plague that came into a lot of these regions that may have killed off parts of the population. But again, we know that the Yamnaya were militaristic. They were highly patriarchal, and other lineages that existed, other paternal lineages that existed in Europe, almost disappear when the Yamnaya come. And then there are a lot of future cultures that are associated with the Yamnaya that then take over Europe, the rest of Europe. So after the Yamnaya, there's this corded ware culture that's from 3000 BCE to 2350 BCE, and they're named that because of their pottery. Then further west in Europe, there's the bell beaker culture, 2800 to 1800 BCE. But these Yamnaya and the descendants of them travel east as well, and they go into Central Asia. So part of the break off from the corded ware is a culture called the Sintashta, and they were in Central Asia, and they end up bringing things down to, and their descendants ended up taking the culture and also the Y-DNA lines into Iranian lands and into India. And so, what's very interesting about the Yamnaya is that they were part of a different haplogroup. So, almost all of the men in the Yamnaya, they were part of R1b. So, not haplogroup G at all, R1b. And within that, they were part of the subgroup called RZ2103. Um, and then with these other cultures, closely associated lines became dominant in these other places, but they all are either part of R1b or R1a. And so today, for instance, R1a is very prominent in R1a was part of the corded wave that went east, and R1a is found in all of the upper class in India, for instance. They all descend from R1a males that come from the corded ware initially that were descended from the Yamnaya. And we also find a lot of R1A in Iran as well, along with the Middle East. R1B, they're different subgroups, but the ones who are directly descended from the Yamnaya stayed in Southeast Europe for the most part and into the Middle East. I can tell you there are 7 different lines of Ashkenazi Jewish lines that are directly descended from this Yamnaya people. They are part of RZ2103, but these 7 lines only constitute about 2% of the male Jewish population. So most Ashkenazi Jews do not descend patrilineally from the Yamnaya. But if you include all of these other associated cultures, you have a much larger percentage of the population. And every person who is Jewish today, who's an Ashkenazi Jewish today, does descend from the Yamnaya in some way, or from these other cultures, but almost certainly from the Yamnaya as well. There's a lot of DNA that is in the Yamnaya in a lot of the Eurasian continent today. And what was so interesting about this turnover in the Y-DNA, too, is that it helps explain the diffusion of Indo-European languages. It's thought that the Yamnaya are these Proto-Indo-Europeans that could also be Proto-Anatolian, too. There's some discussion about how Anatolian languages came to be, but certainly the Yamnaya represented this Proto-Indo-European languages that brought Indo-European languages westward, and then we can also chart how the Indo-Iranian languages and languages like Hindi and Farsi, how they broke off. But they all are associated with the diffusion of these people who migrated initially from the Pontic steppe westward, and then some went further west into Europe, and others went eastward. Okay? And they eliminated a lot of the male lines that existed prior to that. In fact, it was like the biggest turnover in the population. And it wasn't just men, too. We know that also a lot of the women who end up living in Europe, they end up being Yamnaya women and people who came with them, too. So it seems like a lot of the local population, did not survive these invasions by the Proto-Indo-Europeans. So again, pretty amazing that we've learned all of this just in the last decade. There's been a lot of research over the last decade looking into this more, but it's a huge part of human history. It's before written history in this part of the world, but we know that it happened, and it helps explain the diffusion of the Indo-European languages. Maybe I'll stop here before we start talking about R-line, G2B, and GM377. Does anyone have questions before we move on about the Lehmann line? That was, again, a background about an adjacent branch that the Stein family descends from, that branched off in 20,000 BCE. Any questions?
Jeremy, before you go into this, I'm curious just about the distribution of time. You go from like 2500 BCE, I'm talking about our line now,
Okay.
into 800 or CE. The question is, can you get finer in between? Like,
Well,
can we know
yeah.
our line during the time of Tanakh as opposed to before and after?
It's a great question, and the answer is we don't know that right now. So it makes a big jump. We'll talk about the reasons for that, for why, but with our current set of knowledge, there is a big gap in time that we are unclear about. So part of the reason why I wanted to talk about G2A first is we know so much about this history. When we get to our line, it becomes a lot more murky. And there are reasons for it, but it's not as well represented in populations, either in the modern day population or in the ancient DNA population, which makes it hard to get to any conclusions. But we'll get to that. We're going to get to that really in the next few slides. So maybe let's move on with that. So now we're going to get back to our line, to the Lehmann line, the Lehmann paternal line. So, and I wanted to provide that introduction with the adjacent line as a contrast. So GM377, that's the line that we descend from. There are no, and again I want to emphasize that there are no ancient DNA today that have been found that are part of GM377. There's none of that. So in the last 10,000 years, we haven't found a sample that is connected to us within the past 12,000 years to 10,000 BCE. What we have found are two people who are a little more closely connected than those G2A folks. So there are two samples. One dates to a cave in western Iran in the Zagros Mountains, and that's from 7200 BCE. And then there's one other one in northwestern Armenia, part of the Kura Araxes culture, that's from 3200 BCE. These two individuals, though, are not really closely related to us at all. They share a common ancestor from 18,000 BCE, so it's only 2,000 years later, but they're based in the Near East, either in Iran or in Armenia, and they lived, 10,000 years after they broke off from our line. So it's not very helpful, but I wanted to show that we don't have any samples from GM377, and even the ones that are date a little bit further behind than that aren't very helpful. So we have to turn to modern day population testing for now. Now, part of the reason why we may not have discovered anything yet, by the way, is that it is pretty clear, because it hasn't been found in ancient DNA, that our line was never in Neolithic Europe, as opposed to G2A, which really expanded westward into Europe. It was a dominant, Neolithic lineage in Europe. G2B, GM377, G2B1 was almost certainly never in Europe. It was always somewhere in Asia and Western Asia. So why do we think that? Well, we're going to turn to the modern day testers. So where have we seen testers who are part of GM377? Well, I denote here 3 different locations based off of how closely related they are to us. So the closest connection on this slide, we're going to talk about ones that are even more closely connected, but people who share a common ancestor within the past, let's say, 6,000 to 12,000 years, they're found in 3 areas. The first is a tester in Saudi Arabia. He shares a common ancestor with the Lehmann line from 4350 BCE. Then if you go a little bit further back, there's Lebanon and Syria. There are testers in Lebanon and Syria today, and they share a common ancestor with the Lehmann line going back to 6350 BCE. So they're a little bit more distantly connected by a couple thousand years. And then the Armenian connection, they're just 10,000 years ago, 12,000 years ago in 10,000 BCE. So we see all of these, right? These are more connections within the past 10,000 years, and where they live today. It suggests a possible origin of GM377 in the Near East. But for now, it's largely a mystery, because we don't have any ancient DNA samples to place a person who was in GM377 at a specific estimated, date in ancient history. Hopefully we'll be able to find such a sample in the coming years. It's only been in the last 10 years that we've really been working with ancient DNA. So, and a lot of these places where we could probably find sites are in hard to reach areas for scientists, let's just say, geopolitical tensions and whatnot. It's not easy to get DNA samples from this area of the world. Okay, so now we're going to get to a little bit closer to the present day, to 2700 BCE. And I have a map here of our closest non-Jewish relatives. So before we showed some of the more distant non-Jewish relatives from the Near East, who are between 6,000 and 12,000 years, all of these individuals that are found here, all of the descendants share a common ancestor within the past 5,000 years, only back to 2700 BCE. Now, it's still before the advent of Judaism, but these people all are our closest non-Jewish relatives along the direct paternal line. So within GBY 794, which formed around 2700 BCE, we don't know where it formed, but the guess based off of the modern day population is either the Near East, the Caucasus, or the Iranian plateau. Most likely it's somewhere there that in 2700 BCE our ancestor was living somewhere in this area. And there are two main subclads, so there are two main branches that descend from it. GBY 764 and GM283. So first, we're gonna talk more about GBY 764. It's also called GY 12975. It has a couple other names. It's a large Ashkenazi Jewish branch, and the most recent common ancestor for everyone who's in GBY 764 is 750 CE. So this branch, so the Jewish branch, which is the one that we descend from, it exhibits a strong founder effect. So what does that mean? And we've talked about this previously, this term founder effect, with the Reuben line. It's actually very, very similar. So all of these Jews who are part of GBY 764, they descend from this person from 750 CE. But they descend probably from one man who was alive then in around 750 CE. He was a survivor of sorts. He was the founder of the subsequent lineages that all followed before that. Until that time, between 2700 BCE and 750 CE, he was the only person who, who's an ancestor whose descendants survive today, who he has descendants who are alive today. And, between 2700 BCE and 750 CE, there were 65 mutations that formed between those two time periods. So by the time that first GBY 764 man, who all the Ashkenazi Jews descend, he had accrued 65 mutations that all of these other branches don't share, all of the adjacent branches. So that's a marker of time, right? Because we mentioned that mutations form every 50 to 150 years. You can date those 65 mutations as going back to 2700 BCE. And we're sort of in the dark as to what happened during that time. We haven't found a person who shares only 30 of those mutations, or someone who shares only 45 of those mutations. If we would find someone like that who lives in today, or if we find someone from ancient DNA, that could help us understand a lot about some of the earlier ancestry between 2700 BCE and 750 CE. So, a closely related term to founder effect, a founder effect is just saying that there's one person who ended up, founding a new population of descendants, and from one person came a lot of people. There's a similar term called a bottleneck. A bottleneck is a thinning of the population that leads to a founder effect, that can oftentimes lead to a founder effect. So in Ashkenazi Jewish lines, we actually see this quite a bit, where there are founder effects at this time in history. Between 500 and let's say 1000 CE, all of these branches tended to form. There were a lot of Ashkenazi Jewish branches that were formed through a founder effect. And so it's surmised that there was a bottleneck that took place during this period. We know that there were plagues that took place not too long before this, like the Justinian plague. And there may have been a thinning of the Jewish population, and this ancestor who was part of GBY 764, he may have been the only one because he was this, founding of this population who survived the bottleneck of the population. But we're not sure. But we do know that, again, all of the Jews that are part of GBY 764, they descend from this common ancestor from 750 CE. That's our branch. And we're going to discuss that a little bit more in the context of other Ashkenazi Jewish branches shortly. The second major subclad within this, and this is really interesting, honestly, it's very unique for almost any Jewish lineage that's been found around the world. But our, again, closest non-Jewish cousins are people who are in GM283. And they are Pashtuns, so also known as Pathans. They're an Eastern Iranic group in northwestern Pakistan and southeastern Afghanistan. All of the people, so we talked about how everyone in GBY 764, they share a common ancestor from 750 CE. Well, anyone who's in GM283, they share a little bit earlier ancestor. their common ancestry, everyone who's in this box over here, number 2, they share a common ancestor from 50 CE. There are also 2 other groups worth talking about who also connect with us from the same time period of 2700 BCE, but they're just much smaller. So the Pashtuns are a big population that are part of that are our closest cousins. There are some other testers that we found in recent years that who are also as closely connected to us as the big Pashtun population. So the first is there was a Sicilian tester, a tester in southern Italy, who is more closely connected to the Pashtuns. So they share a common ancestor from 1900 BCE and not from 2700 BCE like we do with them. So they branched off a little later, and this individual ended up in Sicily in southern Italy. So certainly that's, Mediterranean region trading with the Near East. That line ended up in Italy. And then we also find someone else also in Central Asia, similar to the Pashtuns. There is a tester from Tajikistan who also represents his own separate line, but also, again, connects with all the others in 2700 BCE. So when you're taking this as a whole, right, the only group that's really in Europe, that's really centrally in Europe, is the Ashkenazi Jewish population. That's us here. We have the Pashtuns, we have the Sicilian and the Tajikistan tester, and it's theorized that the common ancestor would have to have lived somewhere in Western Asia. Again, we don't have any degree of specificity, but it is very interesting to find these connections in Central Asia. It's very unusual for any Jewish lineage to have that as the closest non-Jewish relative. But again, this is all before the advent of Judaism. So I want to emphasize again that all these branches broke off in 2700 BCE from the Jewish branch. This is all before Judaism was formed. So I want to spend a few more minutes about the Ashkenazi Jewish branch that we descend from, GBY 764. And this is really special now that I'm going to be introducing for other Ashkenazi Jewish Y-DNA lineages in the future. So over the summer, I connected with these 2 really amazing researchers, Leo Cooper and Josh Lipson. Leo is going to be an academic in this field, but Josh is is a citizen, very knowledgeable about Jewish Y-DNA. And a few years ago, the 2 of them embarked on looking at all of the people who had tested in the Ashkenazi Jewish population and looking at their lineages and trying to quantify the percentage of the Ashkenazi population that descend from these various lineages. And so they've discovered through their research that there are over 130 Jewish paternal lines that can, in the Ashkenazi Jewish population, that trace back before the existence of the Ashkenazi Jewish community. So GBY 764 counts as one of these lineages because it formed in 750 CE. That's really before what we call the advent of the Ashkenazi Jewish population in the Rhineland. And so, or it's right at the cusp of that. And so it is one of 130 lineages. But what they did when they counted these 130 lineages, they aren't evenly distributed, right? So what I show here on this chart are the top 10 Y-DNA lineages that trace back before the existence of the Ashkenazi Jewish community. And on the left-hand side, I include surnames in our family that are part of these groups. So the biggest haplogroup in the Ashkenazi Jewish population are Ashkenazi Levites. That's the RY2619 branch. Our Lando family were Levim, and they have been tested, and they are part of RY2619. It's almost 8% of the Ashkenazi Jewish population is these Ashkenazi Levites. So when we talk about Ashkenazi Levites, between 50 to 70% of all Ashkenazi Jews who have a history, a tradition of being Levim, they're going to test within RY2619. So the majority of people who have a Levi tradition in Ashkenazi Jewish populations, they're going to be part of this group. So it's very interesting. And at some point, I will present about the Landau family. I've already done a lot of testing in this line. But what is also interesting is that Levim from other parts of Judaism, whether they're Sephardim or Mizrahim, they are not part of RY2619. They have other lineages associated with the Levi lineage. So it's not across different groups of Jews. but it is found predominantly, it is the dominant lineage in Ashkenazi Levim. Okay. Then another one just to point out here is the third one here, JS12192. So this is a Kohen branch. So the number one branch was the Ashkenazi Levite. The third most common branch is this Kohen branch. And that branch is probably one of the most interesting branches in not just Judaism, but I think human history. Why do I say that? Well, this is part of what's called the Kohen modal haplogroup. So we have found Jews around the world, whether it be across Sephardi lands, North Africa, whether Jews in Yemen, Jews in India, Jews in Iran, Jews in Iraq, all across the Near East. In all of these populations, there are Kohen men who descend from a common ancestor from First Temple period in BCE, which is pretty amazing. So there is a Kohen tradition that's existed for 3,000 years that's been successfully passed down. At some point, I want to give a presentation about it because it is fascinating. There's a lot of ongoing research happening here. But by the end of the Second Temple, there were already 60 different sub-branches of this main Kohen branch that ended up in different parts of the world, okay, today. But there were already 60 different sub-branches. So this JS12192 is just actually one sub-branch that ended up becoming dominant in the Ashkenazi lands. But it's part of a much bigger Kohen modal haplogroup. With some early testing, our Kohen family from Metz is part of this haplogroup. So we do descend through our Metz Kohen family through that Kohen modal haplogroup, which is pretty cool. And then I list a few others here. Further down, we've talked about earlier, if you can check out on YouTube, the Reuben family. That's part of EY14891. And then some of these other ones. Hollander is from my mother's side, my maternal grandfather's side. And then the Lichtenberg family is also, that's Grandma Liz's paternal line. They are part of JL556. All of these are top 10 lineages, okay, in the Ashkenazi population, and we have good representation in our family from different surnames. But, the Lehmann line, getting back to that, which is part of GBY764, it's the fourth biggest branch of Ashkenazi Jews. The top 10 constitute about over 50% of all of the Jews, all the Jewish males, these top 10. And we're number 4 in terms of size. And again, we're the second biggest non-Kohen, non-Levi branch. There's only one other branch that's larger, and that's EY6940, which has a very different history than GBY764. But anyways, I thought this chart was pretty helpful just to put in context that there's a lot of diversity in the Y-DNA lineages because there is 130. Maybe this is a good point to sort of compare to Europe, okay? When you talk to Europeans today, there is not this type of diversity in paternal lines. Because of what happened with the Yamnaya and their descending cultures, I think I read like 60 to like 80, at least 60%, maybe up to 80% of Western Europeans' population, they all descend from R1b. There isn't diversity because that whole population got turned over by the succeeding cultures, and there are a lot of different sub-branches and of R1b that exist in these different parts. There's the whole big Germanic branch and whatnot, but they don't have this type of diversity within different haplogroups. So you can see there's R here. This R is definitely descended from, this is an R1A line. But we do see other ones like E and J come up a lot. There's haplogroup G with us. There's also a haplogroup Q. So a lot of much more diversity than what we see in the European population. Okay. So then we're going to move on from GBY764, and we're going to get a little bit closer to the present day. Maybe we'll talk a little bit more about GBY764 and then talk about its subclads. So what do we know about GBY764? So the common ancestor was born in 750 CE. What we also know is that this ancestor and the descendants were among the earliest Ashkenazi Jews to come to Europe. And they settled in the Rhineland over 1,000 years ago. So they were among the early Jewish men to come to the Rhineland. They're among the earliest people to come to Ashkenazi lands. Now, there's again some uncertainty here. I have read and I have talked to some people who say this is real. There might be connections to potentially a Sparta connection that predates that 750 CE. There was apparently some small population of males, Mestizo males in Mexico. This is part of from Merida in the Yucatan Peninsula. There apparently that were also connected to GBY 764. So it could be, and again, more research needs to be done here. If we could test some of these males with big Y testing, it could be found that maybe there's a Sparta connection before 750 CE, but we're not really sure about this. But there might be a Sparta connection with their line, but it's uncertain. Within GBY 764, we're going to move on. There are 4 main subclads. I list them here, but the fourth one, GFT 7446, is our branch. Remember, GBY 764 is one of the biggest Ashkenazi Jewish branches. There are 4 different branches that formed very quickly after 750 CE. I think GFT 7446 formed around like 860 CE. So within 50 years, there were 4 different sub-branches that existed basically, and we descend from this 4th sub-branch within GBY 764. And then this was the final result that I had. So this is looking at June 2024, looking at all the different sub-branches within that 4th subclad of GFT 7446. So you can see up here it says GFT 7446, and these are all of the testers today who are male testers who share a common ancestor from like 800 CE, who are part of this 4th subgroup. So you can see here almost everyone here, except for these Halperns, they all have different surnames. So when you see that, when you have matches that are part of different groups like here, that like Jacobson and Mannheim, Spielberger and Goldbaum, it means that their connections almost certainly predate the advent of surnames. So their connections to each other, even though they're like more closely connected to each other than they are to me, which is over here, they don't descend from someone from the last few hundred years. It has to predate the advent of surnames. Now for us, we don't have any close matches. You can see what we have here. It says GFTA 54806, a terminal haplogroup. That is the last subclad. It's the last haplogroup, the closest to the present day, that you share with another tester. So GFTA 54806, I share with a guy named A. Volterra. So he's our closest match. All these others, connect further back, but A. Volterra, he connects from 1200 CE. He is from a community in northern Italy, and he was tested by this group called Avutenu, which is testing Jewish males around the world. And he had a paternal, I'm not sure if he himself is Jewish, but he had a known paternal Jewish ancestry coming from northern Italy, and he is our closest match. So normally when we look at these big Y results, we can find connections that are closer than 1200 CE. This is 800 years ago. This is even before the Black Plague. It's not that instructive. But what I've learned is that this is not so uncommon with Western Ashkenazi Jewish lines. So you remember our Lehmann family came from Hesse in Western Germany, and the Jewish population in Western Europe, as opposed to the Jewish population in Central Eastern Europe, it's just much smaller. And it's just a much smaller pool of people that descend from them, that have male descending lines, and who have tested. And so because you have a much smaller population that could potentially have taken tests, we don't have a lot of representation from Western Ashkenazi Jewish lines. We added our line to this to learn about that, but there are no more recent testers than that. So that was the findings as of June of this year. So I'm going to stop here and take any questions about the ancient history, and then we'll go to part 2. All right. Okay, so now we spent a long time talking about the ancient history. Now we're going to go into trying to find Lehmann relatives that are alive today and try and get more information about our paternal line by testing them. So, as I mentioned, the initial Y-DNA results, which I got back a few years ago, right? This is back in 2020. It didn't show any paternal line matches within the past 800 years. That was a disappointing result. I was hoping to find things that might be within a genealogical time frame, or at least within maybe like 500 years, something that could be, get closer to the present day. But even though we were part of this huge, Ashkenazi Jewish branch, there are not that many close relatives. So I waited for a while. I basically was just waiting for someone to test to show up on my match list that who could be closer. But as I waited and waited and waited, no one was coming up on my match list. So the years went by and I wasn't really getting any closer. So I decided that I would try and do something myself. And I would go on this quest to find Lehmann surname males from our family who would be willing to take a Y-DNA test. And why did I want to do that? I wanted to, twofold. Well, one, I wanted to learn more about my line and learn about the history of that line. And if we shared a common connection with a Lehmann cousin, we'd be able to see more mutations that we share that accrued, after 1200 CE. The second one was I wanted to also prove through, Y-DNA testing that we are indeed the Lehmann family from Weiderstadt in Germany. But this took a long time. Finding potential Lehmann Y-DNA testers were really, really hard. So this is something that I've seen repeat itself in lots of different surnames in the family tree. And that is, our branch of the family just so happens to have a lot of male Lehmanns, okay? Because my father's one of 4 brothers, and he's got a couple male first cousins, and they have sons of their own. So there are actually a lot of male Lehmanns from our branch that could theoretically test, right? Like I ended up testing for our family, but there were quite a few who I could have asked to test if I couldn't do it myself. As you go back further in time, and chase back all of the family trees, and this again involves a lot of genealogical research, you find that these Lehmann branches died out. So as you go back, it was just harder and harder to find living males who had the Lehmann surname, and I kept striking out. It was very frustrating. But I did find one candidate. And this was back in 2022. I found a tester whose name is Harry James Lehmann III. And he lives in Oregon. And Harry, he descends from, for those who were on the first presentation, we talked about how our ancestor, Simon Lehmann, was one of 4 Lehmann brothers who was born in the 1820s and 1830s. And Simon's 2 younger brothers, whose names were Jacob and Mayer, they moved to the United States in the 1850s. And we actually found the emigration record for Mayer leaving in the 1850s, and he ended up going to Buffalo. So Harry James Lehmann III, he descends from this Mayer Lehmann, okay, who was his second great-grandfather. And so theoretically, Harry James Lehmann would be my 4th cousin once removed. And we share a common ancestor. Our common ancestor would be my 4th great-grandfather, his 3rd great-grandfather, Bunham Lehmann, who we talked about last week. Bunham Lehmann, who was born in 1791, he would be the common ancestor between the two of us. So back in 2022, I asked him, I said, I found him online through my research, reached out to him, reached out to him and said, would you be willing to take a Y-DNA test? I'd love to be able to explore our common ancestry. I've traced our, through the documentation, and I wanted to see, will Y-DNA support this or not? And so after we connected, he agreed to take the big Y-DNA test. So it was really special to be able to have him test. So was Harry Lehmann, was he our Lehmann cousin? And the answer that came back was it was a definitive no. Okay? Harry was not our biological cousin. So what did his results show? Well, he came back as part of the paternal-line J2, major haplogroup, which formed 26,800 years ago. If you recall, our major haplogroup G was from 45,000 years ago. we don't share a common paternal line with Harry James Lehmann going back like 50,000 plus years, okay? It goes really far back that we share a common ancestor. Certainly not within the last 230 years, which is what I was shooting for. Um, so that was pretty frustrating. For those who are interested, I put together a presentation discussing Harry's results and what his Y-DNA line is. It's a fascinating lineage that is also a well-established Ashkenazi Jewish lineage. So he does descend from an Ashkenazi Jewish lineage, but just a totally different one than the one that we descend from. Okay? So when we got these results, it proved definitively that, me and Harry, we don't share a paternal line going back tens of thousands of years. The hypothesized connection was incorrect. And for me, this was a devastating result, okay? Like, I spent years trying to find a male Lehmann, and he was telling me like he knew about a few other potential like candidates, and he's like, oh yeah, this one passed away, this one was adopted. Like everyone else from Mayor Lehmann's branch is like checked off. He was the one person who I could test, and it was pretty devastating. It was like, I thought it was a real roadblock in the research. So I want to discuss what this result means a little bit further on the next slide. So the fact that we weren't a match, it meant that there were inconsistencies between the documentary evidence and the Y-DNA evidence. There was a break between those two. Something was wrong. So what did that mean? The fact that we weren't a match, it meant that either Harry's or my biological paternal line was different than what the documentation indicated. Okay? So we call this that there had to be misattributed paternity somewhere in one of our lines. There's a terminology that's used for people called NPE, non-parent expected or non-paternal event. There was an NPE event that took place in either my line or in Harry's line or both of our lines, because the evidence showed that we were not from the same family. So I took a really close look at Harry Lehmann's results. I mentioned I made a presentation for him. And my tentative, conclusion, if I had to take a guess, if one of our lines was incorrect, my guess is that it probably would have been Harry's and not mine. So I had two big data points that supported this, tentative conclusion. The first was through this other form of mutation testing called STRs, which are different than SNPs, but it's a different type of testing. It showed that Harry had 15 Y-DNA matches from the last 325 years. Recall, I have 0 in the last 800, right? There's only 1 in the last 800. There's 0 in the last 325 years. Harry had 15 Y-DNA matches that were from a more recent period. And where were all of these matches from? They were all from Eastern Europe, from Poland, Lithuania, Latvia, and Russia, right? This is a part of the world that there was a much bigger Jewish population. A lot more people have tested, and it just so happens Harry shares a common paternal line ancestor with these group of testers who all come from Eastern Europe. Well, that's not where our Lehmann family was from, right? Our Lehmann family was from Western Germany going back to the 18th century, perhaps even before then, and I don't think it ever was in Eastern Europe. So the fact that he's connecting with all of these other testers with Eastern European ancestry, that probably indicates that he's from some other line. The second other major point of evidence is that his haplogroup, which was called JY33795, it is not a Yisrael line. It is actually a Kohen line. And this is different than the Kohen modal haplogroup. So it's a separate line of a separate tradition of having Kohen ancestry, but it does go back over 1,500 years. And almost every person who's part of this group, they know that they are Kohanim, okay? They know that they have this priestly lineage, even if they grew up in a less, culturally Jewish household. A lot of them have that. And right, well, I know about our Lehmann family. There's no mention of Lehmann's being Kohanim, okay? There's no connection with, from what I could tell, from our Lehmann line being Kohanim, and but it just so happens that Harry was a Kohen, according to this. So that was also suspect. It didn't seem like it was connected to our Lehmann line, from what the documentation that I had found in Europe didn't suggest that the Lehmann line identified as Kohanim. Okay. But still, despite that, that's not conclusive proof. Okay. That's just like a hunch that it seems to be that there's something amiss with Harry's line, but it could also be that my line there could be issues. So at that point, I looked at autosomal DNA. You can look at DNA matches of other people and see, do these distant Lehmann cousins, do they share some autosomal DNA? It won't be so much DNA, but maybe they'll share some DNA that could hint at a connection through the Lehmann line. And as much as I searched, I had a lot of trouble with that. Now, I could find some things. So, for instance, it was very clear that we descend from Leo Lehmann. Okay, Leo had a brother who had children, and their descendants are my father's second cousins. And guess what? They do. They match. Like, we do have matches from that line. So it was very clear that we descend from Leo Lehmann. But did we descend from people from Weiderstadt from before that? Remember, Leo was born in Karlsruhe, and then you have Krefeld, and then Weiderstadt. So it's a few more generations. Could there have been misattributed paternity in my line as well? So at this point, after Harry's test, there was no answer, okay? Like, there's no way of determining this. And I needed to find another Y-DNA tester from a different Lehmann line to figure this out. Okay. So then the miracle happened, and I somehow found another living male Lehmann from our family, okay? And I call it a miracle because it really was. Like, I was, so many things were involved for this to happen, okay? So back in end of last year, I'm revisiting the different male Lehmann lines. I had put it away for a little while after Harry's test came back, and I was trying to see, is there any new evidence I can uncover that can find living male Lehmanns? And it just so happened that within the past year, the 1950 U.S. Census had been released and had been indexed by Ancestry.com, which is a genealogical website. And as I was searching the 1950 U.S. Census, it just so happened I got a hit on this person I was researching named Alfred Leo Lehmann. He was living in Queens in 1950. And it was a surprising result because I knew that he had been living in England. Okay, he was born in Karlsruhe, but then he had moved to England at some point. And in England, right, you can't get census records that are only 72 or 73 years old. You have to wait 100 years before you can see records. So there are no census records we're going to get from England until the 2050s. But it just so happened that Alfred Leo Lehmann was in the U.S. because he was an interpreter for the UN. He worked as an interpreter for the UN and was living for just a few years of his life. He lived in New York City and was recorded by the 1950 census. And on that census was listed a son whose name was listed as A. David Lehmann. Okay? And I was like, oh my gosh, I have never seen this person before. He's born in the 1940s. Could this guy, A. David Lehmann, could he be alive today? And so, fortunately, he is online. He is a retired professor at Cambridge University. He taught sociology, did a lot of research with Latin America and multiculturalism in Latin America. And again, he's recently retired. But he has a website and, where he has information about his research studies. And so I was able to find him. I was able to find an email address for him on that website. And so I sent him an email late last year. And then I don't hear anything for months. Okay? And then in April of this year, so it's like 5 or 6 months later, I get an email back from this professor, Albert David Lehmann. And he goes by his middle name, David Lehmann. And he said, I'm going to be in New York City. And I'm sorry I didn't write back, but I would love to meet you meet up with you and talk to you about my paternal line ancestry. And so it was like a shocking result that someone would actually write back to me after not hearing from him for several months. So I enthusiastically wrote back, and we met at a cafe in Lower Manhattan a few weeks later. And I shared with him all of this information about his Lehmann paternal line, which he didn't know much about, including the fact he didn't know that his great grandfather was named David. That he's named his full name is named after his grandfather and his great grandfather. We talked about David Lehmann, the ancestor, who was his great grandfather, last week, right? He was apprehended in the November pogrom in Weiterstadt. So his great grandfather was one of the last Jews to leave Weiterstadt. His family stayed in Weiterstadt until at least his great grandfather did, until the late 1930s. His father and grandfather were already living in England, and they helped bring David Lehmann to England before the war started. And so his grandfather and great grandfather, although they died before Albert David Lehmann was born, they died in England. They were not impacted by the Shoah. So anyways, I spent all this time talking about his ancient history, and then I said, I had already bought a Y-DNA test, and I was like, could you please, I would love to be able to research your Y-DNA line and see, right, I've tested one other person, he wasn't a match, you're going to decide it for us. Who is the real Lehmann? Will the real Lehmann, please stand up, and we'll see who you match. Maybe you'll match me, maybe you'll match Harry, maybe you won't match either of us, but it's really helpful to have this other data point because I don't know of any other male living Lehmanns. And at that point, I took a deep breath because I wasn't sure how he was going to answer that, and he said, sure. After like a dramatic pause that seemed like a long time. He said, sure, and the next day, he took the test and submitted it. So really, very, very grateful that he also decided to take the test. And so I waited impatiently for a few weeks to get for the results, and what did it show? Well, it was a very exciting result. Why? Well, it showed that this David Lehmann, the professor, he was from our Lehmann paternal line. How do we know that? Well, our haplogroup, which had been our terminal haplogroup, which had been GFTA 54806, it now became GFT 37500. Okay, and they're actually, we're going to talk about it. There are a set of 6 mutations here that only me and Albert David, who have tested, only the two of us out of all the big Y testers, these 6 mutations are the only ones that the two of us share, that no one else shares. And they are markers of time, right? They're markers of time up to our common ancestor, who was named Bunham Lehmann as well. Bunham is our common ancestor, who's my 4th great grandfather, and his 3rd great grandfather. He had all of these mutations when he was born, and it was then passed down to each of our separate lines. And so our haplogroup became refined, and it was a very exciting result because it showed, we know, right, that Albert David Lehmann was definitely from Weiterstadt. His great-grandfather was one of the last Jews there, and he is a hit. He is a match. He is our closest match, and it's not even close. There were 6 mutations. That's a long period of time between our connection and connection with any other tester in the world. Okay? So let's talk about these 6 mutations. I have them listed here. The top one is what the name is, but it could be any one of these could be our terminal haplogroup. It's not we can't really differentiate between when they arose, but it's one of we have these 6 mutations that only the two of us share. And so what does this mean?
Jeremy, can I ask?
Yeah, go ahead.
Why did you focus on him? I mean, there are a lot of people living with the name Lehmann, right? What indicated that you wanted to focus on him when you found this census thing?
Because of I'll show in the appendix, I do include the census data, but it included the full name of the father, Alfred Leo Lehmann, and it matched the age that I knew that he was born, and it said that he was born in Germany and that his kids were born in England, and it matched, but I wasn't sure.
It matched what you knew.
It matched what I knew already, right? But I wasn't 100% sure. So like my first initial email, I'm like, I think you're from this family. I hope you are. And then he never responded back. So I was like, oh, maybe I'm wrong. Because from all of the writings that Albert David Lehmann, the son who tested, he never really wrote about his paternal line. He actually has talked about his mother's line and his mother's ancestry, but he didn't know much about the Lehmann line, and so it was interesting for him to see all these records about his ancestors that he didn't know about. He didn't know that he had family that died in the Shoah that were closely related to him. There was a lot of stuff that was like new to him from this because his father never spoke about his family. Right, his father's father had died right before he was born. The father's grandfather died also in the early 1940s, and the father never spoke about his ancestry. He just shut it off and never really spoke to him. So he never knew about that side. But yes, what led me to that was that I was not going to just test any random layman person who had the surname, because it's, as I mentioned, like we come from a very small layman family, from a small little rural town. There are a lot of different laymans, and I wanted to do targeted testing. I wanted to have some sort of documented, evidence that supported, testing this individual. But there were very few people. Again, I haven't found any other people who I could test that are distant relatives. And if we tested, let's say, it wouldn't be as informative at that stage to test, my first cousin, let's say, or test you, test an uncle. What that would show is we would see all of the mutations that have formed up until, let's say, Grandpa Henry was born, which is interesting. It's still very interesting, but I wanted to see what did our earlier ancestors, what did their mutations look like? What was their subset of mutations that were passed down? And so what's nice about this, these 6 set of mutations, they came into existence by the time that Bunham Lehmann was born in 1791. We know that because it was passed down to his line, it was passed down to my line, and our common ancestor, he was born in 1791. It could not have arose after that point. So we know that by 1791, these 6 mutations had come into the world. I also, the way that I found out, I actually got confirmation, there is no one else in the world today. This is not even just in our line, right? It could be that independently these mutations could have occurred in some other branch throughout history, and it's just part of a different, totally different subclad, but they also had this mutation. I got confirmation from a senior person from the company that no, these 6 mutations only exist in the Lehmann family as we know it in the world. No one else has these mutations who's ever tested. So that's pretty cool, too, that it's very unique to our line. And so what I say here is that these 6 mutations represent Bunham Lehmann's Y-DNA SNP signature. So SNP is a mutation, so it's the Y-DNA mutation signature. It's if we test anyone, right, who claims to be a male line descendant of Bunham Lehmann, they have to have these 6 shared mutations. Otherwise, they're not descended from him. So again, it gives us insight of what Bunham Lehmann, who was born in 1791, who we found the birth record for, right? Back from last week's presentation, we now know what his Y-DNA looked like and what set of mutations he had that have been passed down since then. Now, what's also interesting is that since that point that our branches broke off, David has 3 private mutations in his line. So that means that there are 3 new mutations that arose in his line in the 5 generations that he descends from Bunham's birth. Okay? So over 155 years, 3 new mutations have formed in his line that are markers of his branch of the family. But in my line, I also have a lot of mutations. I have 4 mutations that arose in the ensuing 6 generations between Bunham's birth and my birth in over a period of around 200 years. So if we do the math, right, 3 mutations over 155 years for David, 4 mutations in my line over 200 years, that's an average mutation rate of 50 years. Now, we talked about how a mutation happens every 50 to 150 years. We're on the really short end of that, which means it actually could be pretty interesting to then do further research, in descending branches, to be able to learn about the other later ancestors and their own Y-DNA SNP signature. So for instance, if, again, a first cousin or uncle were to test, we could see how many of those 4 mutations are shared with me. we could then look at what, Grandpa Henry, who was our shared common ancestor, how many of those four did he share that were passed down to the both of us? How, conversely, I could look at my brother, right, or have my father test and see how many of those are, shared from my father, and which one came, maybe there was one that came with my birth that is unique to me, that is being passed down to my sons and forming these new branches, these new sub branches. But it's not often the case that you see so many mutations in just a short period of time. So it actually, because we're mutation rich, both of our lines, not just my line, but David's line too, because we're mutation rich, we could actually do further studies to research our line and see how some of our closer ancestors, how many more mutations they had, and we could look into that more into the present day. Conversely, we can also use this to look back in time as well. So we have these 6 mutations. Maybe down the road, there might be someone who only shares 5 of these mutations. Or maybe there will be a tester who shares only 4 of these mutations. They will be more closely related than anyone else but our Lehmann family. And they will represent a connection sometime between 1200 and 1791. So anyways, this allows for future research down the road. So I'm just going to summarize here as we're getting close to the end here. So after years of searching for Lehmann male line descendants, it yielded 2 different individuals. There was Harry James Lehmann III and Albert David Lehmann, both born in the 1940s, and they both took big Y DNA tests. Harry Lehmann's results in 2022 proved that he was not a paternal line relative of mine, but David Lehmann's results in 2024 were a match. And this is very strong DNA support that he and me, that we both descend from Bunham Lehmann, from Weiterstadt. And our terminal haplogroup is now called GFT 37500. And this and the additional 5 mutations form the Bunham Lehmann Y-DNA SNP signature. They help us identify additional Lehmann family descendants in the future and can help us with dating connections to other new Y-DNA matches. So with that, I'm just going to get to the thank you slide, and then I'll just highlight just a couple slides in the appendix very briefly. So I want to first extend a big thank you to both Harry Lehmann and David Lehmann for agreeing to take the Y-DNA test. This has been a years-long journey of discovery for me. None of it would have been possible without having these 2 men test. I'm really, really grateful for them testing. I also want to really thank Leo Cooper and Josh Lipson. These guys are of my generation or younger, and they're really impressive people who are helping to expand the field of Y-DNA research of the Jewish population around the world. And their extensive knowledge of Jewish Y-DNA lineages, including those of our Lehmann line, they helped me with making this presentation. So I really want to thank them. And then I just want to quickly, in the appendix, I do have a lot of stuff here. I'm not going to dwell on this too much. This is just diffusion of agriculture. These are all the descending cultures from the Yamnaya and how they spread throughout Europe and Eurasia. But I wanted to point out here this 1950 U.S. Census that really led to the breakthrough. So again, I had this Alfred Leo Lehmann, Alfred L. on here. It mentioned his age, which matched that. It had Germany, and it said that he's translating for the UN. And then this was his son, and he had a daughter as well in 1950. I actually asked David Lehmann about this time because he was, 5 or 6 years old, and he said he remembered living there, and he even remembered this woman, Anna, who was the maid living in their home in 1950. So he thought it was pretty cool to see her name on this record. So again, thank goodness the U.S. releases these records after 72 years and not 100 years, because it allows you to find living people who you could still connect with. And then I just want to get to this. This is a picture of me and our cousin, Albert David Lehmann, at the cafe in Manhattan. And maybe you can spot some resemblance between us. Personally, I think we've got similar noses. I think there's a Lehmann nose that has been passed down through the generations. We definitely have that same nose. But it was pretty neat being able to connect in person with our most distant male Lehmann surnamed relative. So with
And
that, oh, go
does he
ahead.
pronounce it Lehman?
Oh, so yeah, that's a great question. I asked that of him, and he pronounces it the exact same way. Actually, he says Lehman. Actually, yeah. I was worried about with the German or whatnot that it would be something different, but actually he pronounces it exactly the same. So, whereas Harry, he pronounces his last name Lehman, not Lehman.
Yeah.
Lehman. Okay, with that I'm going to stop the stream, and I can take questions.